Our next speaker is Dr. Marc-Andre Schulz from the Charité Berlin. Marc, over to you.
Whoops. Okay. Sweet. Does that work? Yes. Okay. In the last two talks, we've already engaged with this idea of explainable AI to break this veil of black boxness around machine learning models. We've talked about validating these explanations for machine learning model predictions. And what I would like to do is to move on from that to treating these model explanations as data themselves, so kind of leveraging the explanations for further scientific inquiry. So the core idea here is that explanation-derived representations amplify task-relevant, or neurobiological in our case, patterns. So in a first step, let's kind of formalize this a bit and then think about what this concept of an explanation space might actually mean. So just as the input data that we use for our models can be considered as points in feature space, the model explanations can be considered point in explanation space, right? The space of potential explanations that you get given a trained machine learning model. So for instance, if you have a brain age prediction model based on what T1 brain images, and you pass all your data points, all your brain images through the explainer, you're going to get a new data set in brain age explanation space, if you will. So kind of to simplify the maths into words a little bit, the explanation space then is kind of the mapping into the space where the data dimensions correspond to the feature contributions for an individual prediction. That space has a couple of interesting properties, right? It's task specific. So depending on the particular machine learning task that we conditioned our model on, for example, a brain age prediction, we're going to get explanations that correspond specifically to what we asked our model to look for. It's interpretable because in our case, the voxels retain their meaning, right? The input data voxel and the explanation voxel are the same. We can map them to MNI space, and they have a semantic meaning that we're retaining. The dimensionality, right, as I said, is isomorphic to the input space, so we are preserving the neuroanatomical, the spatial relationships, which is going to be useful for analysis. And that's the crucial part. There is, at least that's what we suspect, pattern amplification, right? Ideally, we would expect this space to amplify, to enhance the discriminative or predictive neurobiological structures with regard to the target that we conditioned our model on. So the good thing is that essentially we've transformed our data into a new space in which we can use exactly the same statistical and analytical approaches that we are used to applying in the original image space, like voxel-based morphometry, clustering, statistical analysis, pattern analysis of all sorts, right? So let's delve a bit deeper into this idea of pattern amplification in explanation space. One problem that we have with brain imaging data, signals, T1, resting state data, whatever, is that it is a mixture, essentially, of a lot of different signals, right? Just basic random anatomical variations, age effects, sex effects, and then potentially, let's say, disease effects that we are really interested in. That's a bit of a, how to say, cocktail party problem, decomposition problem, where ideally we would like to isolate the part of the signal that we're really interested in, and to some extent, using this explanation space for a particular machine learning model, let's say a disease classifying model, gives us exactly that, right? It amplifies the task relevant, the disease specific signal, and suppresses the signal irrelevant to the prediction task, the disease classification task, right? So ideally, it should enable us to detect subtle effects in the data that are masked in the raw data, but ideally more visible because of this decomposition in explanation space. Another effect, I'd say, is essentially dimensionality reduction with feature preservation, right? A problem that we have with brain imaging data is that it's very high dimensional. So we have, depending on what we apply statistical, like univariate testing, we have the multiple comparison problem. If we do machine learning, we have the curse of dimensionality. So ideally, we would like to reduce the dimensionality. And to a large extent, we do that with kind of transforming to explanation space, because we are creating sparsity in the image, right? A lot of the voxels, say in the T1 image space, are going to be irrelevant for prediction throughout all the participants, all the input spaces. So we are reducing the effective dimensionality based on which we are going to run our analysis, while still retaining the spatial relationships in the original space, which is going to be useful, right? Then we can capture nonlinear relationships, right? So ideally what a deep learning brain age prediction model does is it captures complex nonlinear relationships in the image that we would not be able to detect or represent or access with simple linear methods, right? So essentially what this transformation to the explanation space through the deep net does is it allows us to use linear statistics on nonlinear phenomena to some extent, right? And in the same vein, we are kind of integrating multi-scale information. So it gives us the ability to, incorporate context, maybe context information from the voxels around a particular voxel in a brain region, that we would not be able to do with just basic univariate statistics. So in the end, what we would hope is that the transformation into explanation space increases our statistical power by kind of focusing analysis on task-relevant variation in the data and reducing multiple comparison problems in high-dimensional data by kind of inducing sparsity and downweighting, suppressing, masking, ideally, the features which are not relevant. And I want to illustrate how this will look like on two studies that we've done, one with a more methodological focus and one with a more clinical focus. So the first one is on subtype discovery. So let's say we have biomedical data, let's say T1 data, and we are building a classifier, a diagnostic classifier for a certain disease, let's say Alzheimer's disease, right? So we have that classifier, we pass all our images through to the explanation space, and the idea is then to cluster in explanation space. So if we were just to take the original imaging data, we would likely get no interesting results because the dimensions of variation with regards to what age, sex, other anatomical variations would be usually orders of magnitude larger than what we're actually looking at. Ideally here, we're gonna get exclusively Alzheimer's disease relevant variation in explanation space. So essentially then in that space, we're asking the question, are there subclasses in the data which have the property that they generate different explanations for belonging to the same class, right? Essentially, that is what a subtype of a disease, a subclass in this sense would be, right? It's within a larger class, clusters of data points for which you can get different explanations or different reasonings for why they should be an Alzheimer's diagnosis, right? Different subtypes would yield different reasons for why they should have an AD diagnosis. So essentially, we check that on a couple of differently constructed data sets, always with two classes, two superclasses consisting of subclasses with a couple of clusters, ran them through, in this case, a random forest classifier, applied sharp explanations, and then ran clustering in explanation space and evaluated the extent to which changing from the original space to explanation space increased our ability to recover these subclasses, these subtypes. And here to the lower left, you see essentially the two first principal components in the original feature space. And then on the next slide, we're going to see the first two PCs in explanation space. So this is what we get, essentially, right? When we transform from feature space to explanation space, we can see, even quite trivially, visually, the extent to which that transformation makes it easier to identify the original subtypes that we had in this case, the disorder or the patient class, right? So essentially, the result here is an improved subtype discovery. So the adjusted mutual information that we have between the feature space or the explanation space and the target increased substantially. That is complementary to other dimensionality reduction methods, other clustering methods, and it appeared to be consistent about the setups that we looked at, right? So that kind of supports this idea that explanation space amplifies disease-relevant variation. It's kind of, as I said, complementary to traditional methods, but most importantly, and I think this is kind of most interesting, there's no need for a high accuracy model in this scenario, right? One problem that we often have in our field is that we just don't get the degrees of accuracy that we would need to actually do something useful with this. And the great thing here is that we really don't need that kind of accuracy. We just need the model to be good enough to yield interesting explanations that we can use to generate scientific insight. Second study, clinical one, was on the effects of psychological stress on brain health, where we know that psychological stress accelerates to some extent disease progression in multiple sclerosis. We wanted to know to what extent that also happens in healthy participants and whether the mechanisms that drive that effect are the same between both groups, right? So here, instead of a disease classifier, we took a brain age prediction model. So the explanation space ran on this brain age predictions, and you see a couple of these explanations here to the lower left in kind of the T1 space and overlaid the explanation space. So what we did is we extracted the brain age predictions using a pre-trained convolutional neural network, extracted the instant-wise model explanations, then analyzed the stress brain health pathway, so the functional connectivity that is most associated with an elevated brain age or a positive brain age gap, and then analyzed the stress brain health endpoints, that is the neurological constituents of this brain age gap in explanation space, right? So how does that look like in the end? We were able to kind of, in explanation space, identify that the same neural pathways moderate increased elevated brain age, so kind of decreased structural brain health, I suppose, in both groups, so both in health and in multiple sclerosis, the same functional connectivity was associated with the elevated brain health, that this is kind of a stress-specific effect, and it has prognostic value. So to some extent, we can predict the disease progression in multiple sclerosis based on this effect. So that's fair, that's just the clinical findings, but the interesting thing is, and that's what I want to focus on here, is what we can then, like on top of the clustering that I said in the beginning, what we can do is we can do interesting linear models on the brain, on the model explanations, right? So in this case, that means that we're looking at regional brain aging patterns. So kind of as a side note, effectively what this gives us here is something like a localized brain age model, right? It kind of takes the brain age prediction and maps it back into the constituent voxels, and we get an idea which voxels to what extent were kind of positively or negatively associated with the elevated brain age prediction. So what we could do here is, again, like run linear models on the on the model explanations, and that allows us to say, for example, for which areas of the brain variation in gray matter fraction were able to explain the elevated brain age in these participants, right? We were able to pinpoint to what, in which areas of the brain the lesion load of participants is mostly responsible for the elevated brain age, and we're able to kind of identify the group differences in explanation space, specifically for stress, right? So again, the interesting thing here is that we get group differences which are specific to the, how to say, in this case specific to the brain age and stress axis, and suppress and downweight all the other dimensions of variation. So again, even though we don't have, how to say, a systematic evaluation or analysis in this case, and compare that to the raw data, we can say we're seeing here effects that, at least in our preliminary analysis, just in the feature space, in the T1 space, we weren't able to detect these effects, and these effects have only become visible to us after the transformation to explanation space. So coming to the end, conclusions, I think based on what we've seen, at least in these two studies, the explanation space does give us an information-dense representation of brain imaging data. I think it's very interesting that there's no need for a highly accurate model, that we can make do with models and that just get us some interesting results, and that the statistical methods that we used to work with just can be transformed to explanation space. But kind of all the downsides and caveats that we've already kind of heard in the first two talks stay relevant and to some extent become more problematic, right? Explanations reflect the model decisions and not the ground truth. They vary depending on how you tune the model. They vary on the specific explanation method. And most problematically, if there's weird biases in the model, they're likely going to be amplified in explanation space. The extent to which you can misinterpret findings, I think, is much higher in explanation space, because again, on top of the general problems with interpreting results, you now have these two extra steps of the model and the explanation methods, which are going to induce their own idiosyncrasies, which you might not be aware of. So I think you need to be, on all accounts, very careful with how you interpret results. Yeah, thanks.
Thank you, Mark. Very interesting talk, really thinking outside of the black box there. You've generated lots of questions, so we'll dive in with one here. The question starts, very interesting talk. If the explanation is used for the results, does the explanation need an explanation? I thought you'd like that one.
I don't know. I'm not even sure what that means.
Okay.
No, I think that requires some thinking on my part. It's probably more of a philosophical discussion, and maybe even more of a semantic discussion as a scientific discussion. So I don't know if I have a good idea on that.
I mean, potentially it comes back to what you were saying about the importance of being careful about causality and the validating of these explanations, really. You know,
yeah.
So
again, like to the extent that the explainability methods are well validated, and we know exactly that the explanations mean what we expect them to mean, then I think we're fine. To the extent that there's any question about that, which at this point there is, I think this can only be used for kind of confirmatory results. We can say, okay, there are differences, there are no differences, but really pinpointing that to the particular neurobiology is kind of tricky and requires external validation.
Okay, great. So another point that quite a few of the questions kind of tap into is, you mentioned, you don't need the models to be perfect. They just need to be good enough to tell you something. Is there a way of kind of quantifying what is good enough? Like how, obviously if it doesn't need to be perfect, but it needs to be better than random. So.
Okay, I'd say it needs to be better than a linear model. Because if you don't beat the linear model, then essentially the effects that you're going to see are trivial and are just what you would get just by linear modeling. And at the point where you notice that your model is significantly outperforming a strong linear baseline, then I think you're kind of capturing the nonlinearities that would make for an interesting profile in explanation space. But I don't have, apart from kind of rule of thumb, I don't have a good idea yet of how to quantify that.
Right, so you sort of use the standard model as a benchmark and show that you're doing better. Yeah, that's a good answer. So another thing that a few questions have tapped into is around the idea of potential data leakage or double dipping, because you've kind of using the same data throughout this pipeline and you're kind of going back into it. Do you need to impose some split in the data, or is it okay to kind of do it in this way?
It kind of depends on the particular research question that you're looking at, right? For the subtyping, for example, there's no association between the labels that the model was trained on and the subtype information, right? There's no mutual information between the class label and the subtype label. So in this case, I think there's like trivially no data leakage. In the second study, the model was trained on external data. So it was a kind of a pre-trained brain age model, and we were careful to not kind of contaminate it with data from the original participants.
Okay, great. Another interesting question here is, so would embedding from a label-agnostic self-supervised learning model be better for downstream tasks, unlike the kind of supervised learning that you've been using? Have you thought about that? Maybe not.
But potentially, I think this, neural network embedding space is different on two accords. One of them is, again, it's not conditioned on the task that you're actually interested in. So there's a very high likelihood that it contains lots of information that you're not interested in. So essentially, you're going to have to start decomposing and extracting the interesting part of the signal again from scratch. And secondly, you are losing the spatial information, right? It's kind of like applying PCA, right? You can take the first couple of principal components, but they are not as easy to interpret, they're not as easy to visualize, and they're not as easy to kind of do other stats on. So you are sacrificing, I think, a lot by using internal embedding spaces from neural networks instead of projecting back into the original space.
Right, yeah, makes sense. All right, well, thank you, Mark. Very interesting stuff. Thank you.